Hello. In this video, we'll cover how to format and upload your abundance or RNA-seq data to Mode. We highly suggest users run their omics data through PMart first and output the expression data, sample information, biomolecule information, and statistics results files from the PMart Downloads tab so their data is properly formatted for Mode. First, specify your input data type. For abundance data, this should be MS-NMR, which stands for mass spectrometry or nuclear magnetic resonance. If this is RNA-seq data, please select RNA-seq. The only required upload file for Mode is the expression data, which can be a CSV or TXT file. In the expression data, one column contains the biomolecule identifiers, and the other columns contain the relative abundances per sample for mass spectrometry, the peak area for NMR, or counts of transcripts for RNA-seq based omics. Here we have a peptide-level proteomics dataset from a mass spectrometry experiment. The peptide column in these data indicates the identifiers for the biomolecule. For other data types, the biomolecule identifier column could contain proteins, lipids, metabolites, or transcripts. The other columns in expression data contain the samples with relative abundance measurements for each of the biomolecules. Next, you may optionally upload a sample information file to specify sample groups. If you do not have this file but would like to specify groups, you may do this in the pre-process step, which you will cover in the pre-process step video. Sample information must have a column with the names of the samples from the expression data file. The other columns in the sample information file could include additional information about the samples, including their grouping information. Here we can see a sample information file for the same proteomics experiment, where one column indicates sample names and the other indicates the group each sample belongs to. Another optional file is the biomolecule information file, which contains additional information per biomolecule. In this example, one column contains the name of the biomolecules and should include the same biomolecules as the expression data file. In the current example, this is the peptide column. The additional biomolecule information in this case is the protein that each peptide maps to. The last optional file is the differential statistics file, which is different for MS-NMR data and RNA-seq data. For MS-NMR, there should be one fold change per comparison and at least one p-value for each comparison, which can be from either the g-test for presence-absence or the ANOVA for differential expression. We highly recommend utilizing the Panomics Marketplace or PMart to get these values. Use this link to reach the PMart application. For RNA-seq data, there should be one fold change column and one p-value column per comparison. For more help on differential statistics, see our video on how to properly format statistic files for PMart in the description. Note that if differential statistics are uploaded, there will be additional settings to specify in the pre-process step. When finished, click Confirm to proceed. In the Preview Tables tab of Mode, you can visualize the uploaded tables to Mode to confirm that the data looks as you expected. Note that the comparison table is relevant to the pre-process step. Also note that if you would like to access the full plotting capabilities of Mode, all 4 files should be supplied. Next to each upload, there is a button with a question mark on it with further descriptions of each file type. Example data are available by clicking on the Click Here for Examples button, where users can download these 4 files for a small metabolomics MS example, a larger proteomics MS example, and an RNA-seq example. See our bonus video titled Using Our Example Data for a thorough walkthrough on how to practice using Mode with an omics dataset.